Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about vaccines. In the vaccine type categories, we have whole pathogen, subunit, and nucleic acid. Today we will talk about nucleic acid. Within that subcategory of nucleic acid vaccines, we have DNA plasmids, we have messenger RNA, and we have recombinant vector. This approach is going to introduce genetic material that will encode the antigen into your body's own cells. Your cell is then going to use the central dogma theory to create the protein that's the antigen. This technique gives a broad, long-term immune response. It's an excellent vaccine stabilizer, and it's relatively easy at large-scale manufacturing. In the picture below, we're looking at a DNA sequence that have been excised from an organism. This organism was supposed to produce this protein that was on the surface of that cell labeled antigen. That nucleic acid DNA fragment was inserted into a plasmid. Now the plasmid is a recombinant or recombined plasmid. This is then shuttled into your cells. Now your cells will contain the recombinant plasmid, and it will therefore produce the protein. The protein is the antigen. The first specific one that we'll look at is DNA plasmid. A DNA plasmid contains genes for the antigen, and it will be shuttled into the cell. One specific example is the SARS coronavirus in 2003. Another example is H5N1, the avian influenza virus. Another example is H1N1, pandemic influenza of 2009. Then the last one is the Zika virus of 2016. All of these vaccines were created using this DNA plasmid methodology. From the selection of the appropriate viral genes to initiation of clinical studies in humans can take some time. With the SARS coronavirus of 2003, it took 20 months to find the appropriate gene that coded for the antigen and to get that to the clinical trials to study in humans. It took 20 months for that to occur. Later on, with the Zika virus of 2016, it only took 3 months. The second subcategory of of nucleic acid vaccines is the ones that are created by messenger RNA. So normally, messenger RNA is degraded rapidly in the cell by enzymatic activities. Some enzymes, such as these exosomes and these decapping enzymes, are the ones that will destabilize or break down the messenger RNA. New methods in creating messenger RNA vaccines have overcome the instability issues. The third subcategory of nucleic acid vaccines is recombinant vector vaccines. This category uses a harmless viruses or bacteria that can be used as vectors to shuttle the genetic material into the cell. The NIAID is an organization that develops recombinant vector vaccines for HIV, Zika, and Ebola.